Good. Okay. All right. So, and then hopefully all the other people as they come in will get automatically put in. All right. So the extra class. Well, it turns out that week 14 or quote lecture 14 occurs on the 3rd of December. And okay, on the 10th of December is where Hanukkah begins at sundown. So I'm hoping that if we were to have that extra class, this that so-called week 15, that I think would be the easiest place to stick this lecture. And then what I would do is I would move things around in terms of having everyone prepared for that final exam. So right now, let's see. Let me go to. Oops, wrong thing. I'm gonna hit cancel. Okay. Close that. Okay. Bring you into full screen. Okay. So right now we're in lecture number 8. We'll be finishing that up pretty quickly today. And then what we do, just move go from the current slide. There we go. What we will then do is move into lecture number 9, the thalamus, and then lecture number 13, the hypothalamus. So let me explain why there was a gap, because when I originally created these things in lectures 10, 11, and 12, what I did was I have these whole series of slides that look at the brain coronally, then look at the brain horizontally, and then look at the brain sagittally. With actually lectures 11 and 12, I have to complete the labeling of those things, where the slide before basically tells you what all of the numbers are. But what probably is going to happen is I'm going to finish the midbrain today. Then I'm going to go into the thalamus, and I'm quite sure I will finish the thalamus, and I may even begin moving into the hypothalamus. But then over the weeks following, what I may then start to do is stick in the brain sections, coronal, horizontal, and sagittal. So you get that a little earlier in the class, and then perhaps move in that last lecture, neurochemical pathways. So the point would then be is you would then have what would be the practical part of the exam, which is the 100 questions, where we basically look at about 40 of them that we've been doing already, looking from the medulla, the pons, and into the midbrain. That would be 40 questions. Then another 40 questions would come from the coronal sections of the brain. Then about 15 questions come from the horizontal sections of the brain, and only 5 questions come from the sagittal, because what you're going to recognize as soon as we get there, we have far, far more information from coronal sections or horizontal sections than we have from extreme parasagittal sections. But so what I would be able to do is front load that, so you're really able to go over that and whatever. And then I can easily schedule those with each of you, the asynchronous part of the exam. So if there is no objection, what I'm basically going to do is say that that makeup lecture is going to happen on Thursday, December the 10th from 9 to 12. Okay, so that should do things. Is there anybody that has an issue with that?
I don't have an issue, but I have a question. So I noticed, at least I had on my calendar, that the exam was going to be on that date. So how would you change the exam? Like, how would you be proctoring at that point? Like, it would be the week following?
The exam quote is scheduled for that date. Yes.
Yeah.
So what we're going to do is we're going to do exactly what we did in the first exam. In the second exam, you would each take the 100 question practicum individually, one-on-one. I would probably, given the fact that everybody finished the first exam, including sometimes chats with each other, within about 35 minutes, I'm going to schedule an hour and 15 minutes probably for that, or an hour and a half for that second exam. I'm not expecting that it'll take that long. But again, we can talk about this individually if there are people who feel they want more time, and I can always adjust the schedule. Then, so then part 2 of the exam, which you would typically sit in the classroom and write out, I am again going to do an open book type of thing, in which I again give you about a 4-day period to finish it. In fact, what I'm basically going to do, something that I adopted a number of years ago, because I basically get tired of people asking me, what's going to be on the final? Is this going to be on the final exam? Is that going to be a final exam? And then when I go through it, it seems like many of the things are on the final exam, and there are questions. What I ended up doing is right around in the class right after Thanksgiving, I will give you a pool of questions. So there'll be a series of 20-point questions, a series of 15-point questions, and a series of 10-point questions. I will then, you will then have access to that pool. You will then know the final exam comes from those questions. I will then pick a subset of the questions that you actually have to answer, okay? And so what that allows you to do is prepare in advance, but then when you get it, boom, you go and you answer that. And again, I think as we saw in the part 2 open book, there was a lot of material, and whatever, I hope all of you used some degree of active learning. I looked at the stuff that you did, and it seemed like you didn't just cut and paste things and everything. I think by and large, everybody really tried to do a good job, because the whole point is to have you retain this information. So, so again, we will do that last synchronous lecture on December the 10th. Okay, so with that, I'm going to move on with the midbrain. And in the last class, what we were doing was identifying midbrain structures in which one predominant group in the tegmentum were 2 major pathways that contain dopamine. And those 2 major pathways are the substantia nigra, and then along the midline, a place called the ventral tegmental area, or VTA. So what we can basically see here is, if you remembered when we looked at the pons and the medulla, what we saw was 7 nuclei that were identified that contain the neurotransmitter norepinephrine. And norepinephrine is a furtherance of a metabolic, of a synthesis pathway that also creates dopamine. The only difference between a norepinephrine containing cell and a dopamine containing cell is the norepinephrine containing cell also contains an enzyme called dopamine beta hydroxylase that changes dopamine into norepinephrine. So we use these classic numbers that Dahlstrom and Fuchs used, A1 through A7, and moving from the most caudal part of the brain up into the rostral pons. And we saw where all of those were. Then now when we move in the midbrain, what we have is another two major catecholamine-containing pathways. The A9 cell group, which is the substantia nigra, and the A10 cell group, which is the ventral tegmental area, or VTA. Now, the point is as we saw with, first of all, when we looked at something like the RAF-A with serotonin, we saw that only there was only a small, not all of the RAF-A cells contain serotonin. Some of them were non-serotonergic. The same thing happens with these dopamine-containing cells in the midbrain. Only about 50% of substantia nigra pars compacta cells contain dopamine. The other 50% are non-dopaminergic. And what's very interesting about that is that the vast 99% of the cells that contain dopamine in the substantia nigra use this pathway that goes from the substantia nigra up to the basal ganglia, specifically the caudate and putamen. And this number one pathway is the nigrostriatal tract, okay? Now, the ventral tegmental area, or VTA, gives raw, again, 50% of the cells are dopaminergic. And the dopaminergic projections going up to the, go up to two different places. One is there is a projection from the VTA up to the limbic system. And shown here is only one of the limbic nuclei, the nucleus accumbens. What we're going to see is a lot of other limbic nuclei that we'll talk about later that receive dopamine projections, like the amygdala. So this number two pathway is then called the meso, M-E-S-O, hyphen limbic, L-I-M-B-I-C, dopamine pathway. Then a third pathway, basically that's dopaminergic, also comes out of the VTA, and it projects all the way up into the frontal lobes. If you remember with serotonin, most of the ascending serotonin pathways that went up into the cortex only were in the frontal lobe. And again, with dopamine, this projection from the VTA up to the frontal lobes is called the meso-cortical DA system. So the major innovation of dopamine in the cortex is in the frontal lobe, versus norepinephrine, where both the dorsal and ventral, but particularly the dorsal noradrenergic pathway that emanates out of the locus coeruleus goes to all four cortical lobes, not just the frontal lobe. Okay, so that is the dopamine pathway. We'll talk about this when we go further. So now a very interesting thing having to do with midbrain dopamine is an issue having to do with Parkinson's disease. And basically here, what we're looking at is L-DOPA uptake. L-DOPA is a precursor to dopamine, and it is incredibly important for the correct activity of dopamine to work. So if you basically look in a normal brain, and right here you can see the basal ganglia. In fact, if you remember here is the anterior horn of the lateral ventricle. And what did I basically always tell you about the lateral surface of the lateral horn of the anterior horn of the lateral ventricle? Wherever you see it's like Mary has a little lamb. Wherever you see the lateral surface of the lateral ventricle, you will see the caudate. So you see the caudate nucleus here and here, and you can see further out the putamen, okay? So the caudate and putamen, collectively known as the neostriatum, what you can see is an intense amount of dopamine in these regions, okay? And you can see it's intense, which means that there's full innervation, etc. Now you compare that with L-DOPA uptake in a Parkinsonian patient. First of all, so what is basically happening in Parkinson's disease? What you're basically doing is losing the ability of these dopamine-containing cells to produce and release dopamine. And part of it has to do with the whole issue having to do with this precursor, L-DOPA. And the point of the matter is, this became what? The major type of therapy for Parkinson's disease. That is treating people with this precursor. And you can see in an untreated patient, basically in this particular patient, the only amount of substantial normal dopamine, brain dopamine that's happening here is in a very small part of the caudate. And you can see the putamen is not working, and you get very, very sort of diffuse types of activity, which basically in some ways starts to argue that there are basic changes, not only in the amount of dopamine that's being released, but how it gets released. Here in an intact organism, the substantia nigra is really pumping out dopamine towards a very directed place, the neostriatum, which then, of course, in turn controls the basal ganglia that then controls all types of extrapyramidal movement. Here now, not only is this dopamine response less in a Parkinsonian patient, but it also appears far, far, far more diffuse, basically arguing there may be some types of very ineffective ways of the brain reacting to the fact that the brain dopamine levels, especially in the substantia nigra, are dropping and dropping and dropping. Okay. So for a fairly long time, the major animal mechanism by which one could go and study an animal model of Parkinson's disease was to, in some way, shape, or form, block dopamine transmission or reduce the ability of the cells to release dopamine. So for a lot of studies, what would basically happen is you would give continuing doses of neuroleptic drugs like chlorpromazine, otherwise known as thorazine, or haloperidol, otherwise known as Haldol. And what you would basically get is this sort of stoic type of how you would see the patient. The patient would be very, very flat affect, basically just sitting there, hardly moving. And this is because of very high doses of neuroleptics, which would inhibit movement. And probably again, I reach back to popular culture. And if there is a place where you basically can see neuroleptics at work in a movie, is a famous movie based on Ken Kesey's novel called One Flew Over the Cuckoo's Nest, in which the protagonist, this guy magnificently played by Jack Nicholson, figures he's going to have a jail sentence, and rather than go to prison, what he basically does is he feigns mental illness, and basically what happens after that is he's put into this mental institution where he really tries to game the whole system, and then you just see what basically happens. But in many of the patients, you're seeing patients with very high doses of neuroleptics. Then, of course, in the early 70s, there was the discovery of a catecholamine neurotoxin called 6-hydroxydopamine that I talked about in the last class, or 6-OHDA. And if you give 6-OHDA along with another set of drugs, what you can do is very selectively damage dopamine nerve terminals and destroy them. But again, these weren't the best types of drugs, especially if you wanted to affect, especially the substantia nigra. And then all of a sudden, in the early 80s, a clinical finding done by a guy by the name of Langston out in California, in University of California, San Francisco, all of a sudden a serendipitous finding happened. Basically what happened was they had a sort of free clinic in the sort of post-hippie type of generation around Haight-Ashbury and whatever. And in the early 1980s, Langston was serving there as a neurological resident. And all of a sudden, something like, I think it was 6 different people, all in their early 20s, all came into this clinic. And what they were manifesting was extreme Parkinsonian-like symptoms. And the reason, what basically found was that all of these people knew each other. In fact, a number of them lived together in like a little commune, and all of them were using or abusing opiates. And what basically happened when he did a little bit of epidemiology, he realized within a few weeks before these people presented themselves as a group, they all had tried this particular form of designer heroin. And so he basically looked at this, and one of the things that was pretty clear was that it wasn't the heroin that was producing the Parkinsonian symptoms. There is some relationship because opioids and dopamine have close interactions with one another, but that wasn't the case. It was actually the substrate in which this designer heroin was there. And what this designer heroin, it turned out, to contain is a substance called MPTP. It is a very, very long name. You can Google it. You can see the full name of MPTP. But what MPTP does is, as you can see here, it's found out that MPTP basically is readily uptaken by astrocytes. So MPTP comes into an astrocyte or a glial cell right next to a dopamine neuron. And it then forms into MPDP plus, and finally MPP plus. And then MPP plus is released by the astrocyte and taken up by the dopamine transporter in a dopamine containing cell. And that MPP plus then goes into the vesicle that contains dopamine molecules and basically destroys those vesicles. So basically what you have is this drug that basically goes in and basically does the same thing as the slow process of Parkinson's disease, where you destroy the dopamine containing vesicles. So now when this monoaminergic neuron in the substantia nigra now fires and tries to release dopamine into the synaptic cleft in the caudate and putamen, virtually no dopamine is being released. And the people show this incredible symptomatology.
Professor?
Yes.
Can those vesicles be regenerated or regained in some way?
Can those vesicles be what?
Regenerated?
No, because you're destroying the presynaptic neuron eventually. It has more things, but what it basically does is attack dopamine vesicles. So what you're also doing is, in effect, destroying or depleting all of the necessary enzymes that are found in the bouton to produce dopamine. So all of those things basically go away. Okay, so now the interesting thing with this paradigm is one of the ways that you can study this very, very clearly is this was done especially in monkeys. So by the way, this is an interesting drug because there is a phylogeny to it. Usually when we have a particular drug that affects a particular transmitter or something, it usually affects it across the phylogenetic scale, okay? But for MPTP and the production into MPP plus, it has very, very powerful effects on humans and then our closest relatives, primates. Then as you go down the phylogenetic scale and you get into rats and mice, what you need is a far higher dose of the MPTP to do this like-minded effect. So in a number of ways, this Parkinsonian model, where you could go and study Parkinson's disease, was done, the best work was done in primates. And the way you basically go and do this is you give the MPTP unilaterally, okay? You're only giving it into one half of the brain, not the other. So now what you end up with is a animal subject that is hemi-Parkinsonian, basically on the opposite side of the body from where you did the injection. And you can do chemical analyses afterwards and see a selective depletion on one side of the brain, but not the other. And so these monkeys would demonstrate using themselves as a control the degree of the Parkinsonian deficit that you could go and study. So now, in fact, one of the people that I described when I talked about the monoamines like norepinephrine, dopamine, and serotonin, a guy named Kjell Fuchsa, and then Anders Bjorklund, who did that third very large study, started to move into another direction. What they basically started to ask after they identified these pathways is, what are these pathways for? And then also, how can you, with the depletion of dopamine or depletion of norepinephrine or depletion of serotonin, how can you put this back? And one of the things that people moved into is a whole thing having to do with transplantation. So it's very interesting that in the initial work that was done was the fact that dopamine, yes, is produced by the brain, but it is also produced by the adrenal medulla. And we're going to learn about this when we talk about the hypothalamus and the hypothalamo-hypophyseal stress response and how it then interacts. There's two stress responses. There's a stress response mediated by the pituitary gland releasing ACTH and acting on the adrenal cortex. And then there is a sympathomedullary response that comes from the noradrenergic cell groups of the locus coeruleus and the nucleus tractus solitarius, descending down into the pericanular gray and intermediate zone of the spinal cord, and then where that goes out to the sympathetic ganglion, and then the sympathetic ganglion then modulates the release of dopamine from the adrenal medulla. And basically the way I show this is that it is a pyramid. So the question is, how much dopamine do you need released in the brain to cause something versus how much dopamine do you need to be released by the adrenal medulla to produce effects? And what you find is that teeny tiny amounts of brain dopamine released in the brain produce far greater effects than the amount that's released by the adrenal medulla. So it's a cascade, and this is how things get amplified. So in any case, early in the game, when people started to think about transplantation as a strategy for the treatment of Parkinson's disease, is one of the first things that people started to look at was adrenal medullary transplants. Now, this is tricky because the adrenal medulla not only will synthesize and release dopamine, but it may release and synthesize a number of other substances. And I'll go into that later. But so you have that chance of these other things if you stuck it into the brain, whether or not they would work. In addition, when you're basically taking out adrenal medullary cells, you have to be very, very careful because sitting right next to it are adrenal cortical cells that, of course, release either the human cortisol or the animal corticosterone. And of course, these powerful adrenal steroids have enormous effects up in the brain, which is why we have a blood-brain barrier and why only a very, very small percentage of any released adrenal steroid ever enters the brain. we see some of this is when the blood-brain barrier starts to break down, like in late Alzheimer's disease, where now all of a sudden areas of the brain, like the hippocampus and the cortex, start to get bombarded with higher levels of adrenal corticosteroids, which then basically change the whole synaptology and function. And a lot of this is attributable to all of the cognitive deficits that one sees in Alzheimer's disease. So in any case, in some early studies, what the Swedish group and a number of other groups demonstrated is that if you took adrenal medullary cells and transplanted them into right around the caudate and putamen of the basal ganglia, these cells would become viable, they'd stay in the brain, and they could release dopamine. And what you could see in those animal models is that this Parkinsonian-induced animal now starts to show better responsivity. Not great, but better. And it was very interesting. There was one country that sort of jumped the gun on this and went right into human studies. And this was the country of Cuba. And Cuba had a whole group of Parkinsonian patients who hardly could move, and it gave them adrenal cell transplants. And one of their demonstrations as to how effective this was is they would show these people would, because a large amount of the Cuban population, right around the time of a sugarcane harvest, would go out and harvest the sugarcane. And here they're showing you pictures of these folks harvesting sugarcane, whereas a month before they could hardly move. However, the longevity of the effect and the viability of the effect and all of the side effects, especially having to do with the introduction of corticosteroids, made this a very unreliable way of transplantation. So, of course, in the 1990s and beyond that was the whole thing of fetal stem cells. And that fetal stem cells can become pleiotropic. That is, what they can basically do is you can take a stem cell with various types of factors and literally take that stem cell and now make it like a dopamine cell. And what you now do is with the fetal substantia nigra cell transplants, you take them and line the caudate and putamen in a Parkinsonian animal, and all of a sudden these animals demonstrate viability. Okay, and this then moved into human research, and particularly in Sweden and the Soviet Union, in England, and then finally in the United States. And again, last time I told you that one of the former people taking this course was this guy named Jeff Kordauer. And you have a very, very interesting story here, because Jeff Kordauer, in running the Center for Brain Repair at Rush Presbyterian, supervised the stem cell transplants of these dopamine transplants in the caudate and the putamen of Parkinsonian patients. And these people largely recovered and were doing things. One of them happened to move to Florida, and he was doing very well. In fact, what happened was he had a lifelong love of golf, and he was out on a golf course. Then for a reason totally not having to do with the transplant, all of a sudden a thrombosis comes out of his leg, goes up to his heart. He has a massive heart attack and he dies. So his wife is contacted and she comes out and then all of a sudden yells, oh my God, my husband's brain. Because what they basically did was promise that if the husband ever died, that they would donate his brain to basically look at the post-mortem efficacy of these stem cell transplants. And so what basically happened was Jeff got the brain tissue, basically looked at the caudate and putamen, and basically found that it's not like the little stem cells were just sitting there and they were like little passive generators of releasing dopamine. These stem cells actually started to form axons and axonal things that did right apposition. They made appositional connections to neostriatal cells. So what you basically are doing there is demonstrating a very tight correlation between the anatomical effects of the stem cell transplant and all of the functional effects. And of course, this was the first study that was ever done with that, the first patient that they looked at, and it got published in the New England Journal of Medicine. But what you can basically see here in this a very singular model is this type of an effect. Okay, so here we have the ventral tegmental area with the mesolimbic and mesocortical structures. So one of the other things here that is interesting is, of course, with the idea of a mesolimbic and mesocortical dopamine system, the dopamine itself and the ventral tegmental area was deeply implicated in the pathogenesis of schizophrenia and also in the whole thing having to do with drug addiction. So one of the things that you can see down here is a VTA neuron with its vesicular release of dopamine. Basically, here is the transporter working, where what happens, 95% of the brain dopamine that is released is usually reuptaken for metabolism and then resynthesis. It becomes a very efficient system. Now, what's a way of increasing the amount of dopamine in the synapse? Well, one way that you can do is you can increase the release, which is something like amphetamine does. But what you can also do is you can increase the availability of dopamine in the synapse by giving a drug like cocaine. Because one of the major effects of cocaine is it doesn't necessarily mimic dopamine. Its major effect is by blocking the dopamine transporter. So now whenever there's release, that release in a synapse lasts a lot longer. Now, an important thing about cocaine and very chronic cocaine use is eventually what's going to happen, because there isn't the transporter working normally, the amount of dopamine available for vesicular release eventually goes down and down and down, and then it gets depleted. So now all of a sudden you have a massive crash. Okay. So now we move to the rostral midbrain, and we're looking at, we'll be looking at this rostral midbrain site. and looking again at the cerebral aqueduct, the PAG. And then we're going to be looking at three structures that you should say to me, Bodnar, you lied to me. You basically said that there's a thing called the diencephalon, which is made up of the thalamus and the hypothalamus, and something called the mesencephalon. Yet, here we're in the mesencephalon, and you're telling me that in the rostral mesencephalon, there are three thalamic nuclei, the medial geniculate body, the lateral geniculate body, and the pulvinar. Well, there are, and they are called accessory thalamic nuclei, simply because the area of the thalamus that we have, that is the thalamic nuclei that we define as the diencephalon, is not large enough to accommodate the medial geniculate, the lateral geniculate, and pulvinar, so they're found in the caudal portion in the rostral midbrain, and we'll see that. Then we're going to see the raphe linearis, the red nucleus, then a tract called the fasciculus retroflexus, then a substantia nigra, and a cerebral peduncle. So here is my rostral midbrain slide. Now, in my rostral midbrain slide, the one that we saw a little bit caudal to it was what we called the Mickey Mouse slide, because we had the sort of little ears sticking out, which was actually some of the accessory thalamic nuclei. I go to Walt Disney again, and I call this my Dumbo slide, simply because what we're basically looking at here is the very large pulvinar, and then we're going to have the lateral geniculate body and the medial geniculate body. All of these are sitting out here. Then, of course, we have the superior colliculus. And what you can see is the superior colliculus, otherwise known as the optic tectum, is getting what? It is getting input from the ganglion cells of each eye, okay? And again, about 5% of all of the optic fibers are running up into the superior colliculus. A very small amount also runs into the suprachiasmatic nucleus, and then the vast majority of them run into the lateral geniculate body. So here are these ganglionic geniculate fibers, and then there are geniculo-occipital fibers that leave the lateral geniculate body. So we have the superior colliculus, and above here we have this back and forth is the posterior commissure. Then, of course, we have the cerebral aqueduct with the periaqueductal gray. Then the raphe linearis. Then we have the red nuclei. Then we have the substantia nigra pars compacta, the substantia nigra pars reticulata, and then the cerebral peduncle. So we can see all of these in this actual slide right here. So when up on the top, of course, we have the superior colliculus, which is not lettered. The O is the periaqueductal gray. N is the cerebral aqueduct. P, all the way down along here, is the raphe linearis, that rostral raphe nucleus. Then what we have out here is D, is the medial geniculate body. So if you remember what I told you before, when we learned about an auditory pathway, and you read, oh, and the lateral lemniscus comes into the inferior colliculus in the caudal midbrain, And then the brachium of the inferior colliculus travels up into the medial geniculate body of the thalamus. It sounds like it's going a long distance, but we can tell just a few slides back, we had the inferior colliculus. So here is the medial geniculate body that is getting those fibers from the inferior colliculus in tonotopic fashion. And then they, in turn, through the temporal radiations, those axons go to the superior temporal gyrus. So now, lateral to the medial geniculate body, obviously, is the lateral geniculate body. And that's marked by the letter B right here. And the lateral geniculate body is incredibly laminated or layered. It has 6 distinct layers. And we are soon, when we go into the thalamus, recognize what the importance of those 6 layers are, especially to binocular vision. Then further out here, what we can see on this side is the pulvinar. Okay, so we saw the raphe linearis, and now what we see is the red nucleus, which are the cell bodies of the rubrospinal tract. And what, of course, is going to be happening is that the corticobulbar tract is going to project onto the red nucleus and modulate it, and then outputs of the globus pallidus, that third part of the basal ganglia, is also going to have a large amount of projection onto the red nucleus. Then what we have for T is the substantia nigra. And then what we have out here in UV, W, X, Y, X and Y, and then Z on the other side is the cerebral peduncle. And all they're basically showing you here is how the cerebral peduncle or corticospinal tract is organized as to the part of the body that it's eventually going to project to. So one last pathway that we have right here that is quite distinct is called the fasciculus retroflexus. Now, what we're going to learn when we go into the limbic system is we're going to see a lot of limbic nuclei that sit in the telencephalon, and we'll go through them. And of course, the limbic system is intensely involved in motivation, and the limbic system in turn gets all of this input coming in from serotonin-containing neurons in the raphe, from dopamine-containing neurons in the ventral tegmental area, and then noradrenergic neurons coming from the locus coeruleus and other noradrenergic nuclei. So they are modulated. For a very long time, there is an argument that there is a second set of cells that they basically call the dorsal limbic system. And that is a group of cells that's not pictured here called the habenula nuclei, H-A-B-E-N-U-L-A. You can Google it or look it up in your textbook. And basically, the fasciculus retroflexus is a way by which habenula output comes, and the fasciculus retroflexus allows this habenula pathway to interact with ascending dopamine pathways in either the control of movement through the nigra or through the control of motivation with the ventral tegmental area. So now, the last picture is something basically trying to show you the sort of limits of the, here is the rostral midbrain. So in this, in this rostral midbrain, We can see the superior colliculus. We can see very nicely the posterior commissure. Remember, it goes back and forth, crossing. We have the cerebral aqueduct, and we have the periaqueductal gray. And then what we have is, again, still a little bit left of the lemniscus, medial lemniscus. We have the red nucleus. We have a very large substantia nigra and ventral tegmental area. And then we have the cerebral peduncles. And what you can see here is you can see the medial geniculate body, the lateral geniculate body, and then this overlying very large pulvinar. But then what do we also begin to see? What we now start to see is right up in here is things like the perihippocampal gyrus, the inferior temporal gyrus, then the middle temporal gyrus, and the superior temporal gyrus. So what we're all of a sudden starting to see as we go more and more rostrally, the subcortex is a much smaller area per se than the overlying cortex. So it becomes very hard to study individual nuclei individually on these brain sections. So this is why what I am now about to do is to go and launch into into a series of mini lectures. So what we're basically going to do when we consider the telencephalon is we're going to consider it in chunks. And these chunks that you're going to realize work somewhat by themselves, but then work in conjunction with other chunks. And our whole idea is to slowly begin to build this interacting aspect of the brain. So we're going to be looking at the thalamus. We're going to be looking at the hypothalamus. We're going to be looking at the limbic system. We're going to be looking at the basal ganglia. And then we're going to be looking at the cortex and then the cerebellum. And basically study each alone in terms of its interconnections or independence from one another, and then all of the other connections that came before and is what is going after. So now, let me just look here. I see I have some chats. So, okay, my favorite movie with Nurse Ratched, yes, and the book is so good too. Okay, very good, people. Okay, very good question. Which one is the LGB and which one is the MGB? Okay, the lateral geniculate body lies lateral to the medial geniculate body, or conversely, the medial geniculate body lies medial to the lateral geniculate body. The easiest way to see the difference is whenever you look at the lateral geniculate body, you can't help but notice the laminations, whereas the medial geniculate body sort of looks like a lot of other nuclei sorted together. You don't have striations. So think of the lateral geniculate body not only as being lateral, but more striated. Okay? All right. So now, we go to the thalamus. So again, I am not sure how much in all of your learning about the brain and any other previous course that you've ever taken, how much you know about the thalamus. But very often, what we can do is one word associations, okay? And we say a word, and basically a lot of times the most common word that comes back is what people think is a very global kind of view. So when I said this before with cerebellum, very often over many, many classes, people talk about coordination, okay? And then when we go into the cerebellum, we can see the underlying thing. So there, the name of the structure was very much tied into an eventual type of function, although that function is somewhat simplified. However, whenever you use the word thalamus, and you can see where the thalamus sits, it sits right into the center of things right there. Very often, people are not necessarily talking about function, but why you have a thalamus. They basically talk much more about what the thalamus does. And the most common word that comes up with thalamus is relay, okay? So now, unfortunately, we could not have the Olympics this summer. God knows whether or not we're going to have the Olympics next summer. But every 4 years, you watch the Olympics. And of course, my favorite things of the Summer Olympics is always watching a lot of the track and field events. And one particular team type of work is the relay. So you have the 400 relay, the 1 mile, the 1500 relay, et cetera, et cetera. And so basically what you have here is not the demonstration of something by one single runner, but rather a team. And very often at the end of the thing, they are going to have the 4 people on the winning team, especially if it's from the United States. We're incredibly jingoistic there. What you do is you bring the 4 people together and they get like an interview by a commentator. If you watch these things, or you go back and can probably YouTube it, go and look who the commentator usually interviews. They usually interview the last runner, the person that comes across the finish line, and second most often, the first runner, the person who got the team off to a very good start. In the immortal words of that sad sack comedian, Rodney Dangerfield, the second and third runners of that relay team don't get no respect. Of course, if the second and third runners didn't do their jobs, forget about whatever lead the first runner got, the 4th runner would never be able to make it up. But very often, because of the way that the thalamus is organized with some of its nuclei, basically it's thought that it takes input from place X and then passes, and the thalamus is place Y, and it passes the information to place Z, with X and Z being the beginning and the ending of that pathway, and the thalamus is merely a passive relayer of information. What I think you're going to see here in this lecture is that this is certainly not the case. In many types of cases, it is a powerful integrator of information. And we're going to see that in terms of the intrinsic types of connections that each of these thalamic nuclei have, and what they're communicating with. So this is a classic old textbook, Malcolm Carpenter, who was the head professor of neuroanatomy at the Uniformed Services Medical College. And he wrote some magnificent textbooks. Now, I happen to be using this model. And what we are basically talking about here is 10 thalamic nuclei. However, very often you can think of it as 11, but what Carpenter does is bundle together the VPL thalamus and the VPM thalamus. So in the thalamus, what we basically can see is that there is a dorsal surface to the thalamus and then a ventral surface to the thalamus. And this dorsal and ventral surface are sort of divided by what we call these interthalamic adhesions. It's actually a pathway that communicates intrathalamic and extrathalamic input into the areas. So we're going to talk about 4 dorsal tier nuclei right here. And then we're going to talk about 6 or quote 7 ventral tier nuclei. So the interesting thing is that when folks began to use this terminology, they're basically talking about the thalamus in terms of its relation to one another. And all of a sudden, the terminology started to get tongue twisting and confusing. So let me very quickly go through them. This thing called the anterior nuclear group is actually called the anterior ventral nucleus of the thalamus. Then we have the dorsomedial nucleus of the thalamus. Then we have the lateral dorsal nucleus of the thalamus. Then we have the lateral posterior nucleus of the thalamus. Then in the ventral tier, we have the ventral anterior nucleus, the ventrolateral nucleus, then of course our well-known ventroposterolateral, ventroposteromedial, and then we have the 3 accessory nuclei, the pulvinar, the lateral geniculate, and the medial geniculate. So the easiest way to really understand and get rid of tying your tongue is to begin to use initials. So here are the internal medullary laminae of the thalamus, which brings in intrathalamic and extrathalamic inputs. So what you can see is that the dorsal tier of the thalamus can be segregated. By these internal medullary lamina. So this first one, prominent one, which is sort of sitting very rostrally and it is sitting very superiorly, is the anterior ventral nucleus of the thalamus, otherwise known as the AV nucleus of the thalamus. Now, one thing we're going to see this whenever we go into the coronal sections of the brain, when we go into the horizontal sections of the brain, we basically can see the AV nucleus of the thalamus standing quite separate from the other surrounding nuclei, whether you're going rostral caudal or you're going superior inferior. And it's because of this, not only this internal medullary lamina, but also because it receives fibers from two major tracts, the mammillothalamic tract and the fornix. These are very large tracts, so they really, together with the internal medullary lamina, separate the AV nucleus. So very often, if you're looking at either an fMRI image or something like that, and you can easily identify the AV nucleus of the thalamus. Then the next very large and largest nucleus in the anterior tier is the dorsomedial nucleus of the thalamus. Here you can see it sagittally, and here you can see it coronally. So it sort of takes up the whole pretty much center of the anterior portion of the thalamus. Then we have two rather small nuclei, which are embedded within the internal medullary laminae. So the dorsomedial is called the DM, the anterior ventral is called the AV. The other two that are intrinsic thalamic nuclei are the LD and the LP, okay? So now we move to the ventral tier. And what we can see are three very large and prominent ventral tier nuclei. The VA, or ventral anterior, the VL, or ventrolateral, and then the combined VPL and VPM, ventroposterolateral, ventroposteromedial. And now we then move to the very caudal portion of the thalamus, which actually goes into what we were considering the midbrain. One of the largest thalamic nuclei is known as the pulvinar. And then you have two of the geniculate bodies, the medial geniculate body for audition and the lateral geniculate body for vision. So now in this Carpenter drawing, by the way, you can go to 40 human neuroanatomy textbooks. Sometimes you might have 8 nuclei, sometimes you might have 15 nuclei. It depends on how people divide things up. I'm just using this as a model. Okay, so now, let's now look at not only the name of the nuclei, but now what is the, where did these nuclei go, and what is the major inputs? So of the AV nucleus, and we're going to go through this individually right now, after I finish with the overall, is we can see the AV nucleus of the thalamus receives input from the mammillothalamic tract and the fornix. And the major output of the AV nucleus is going to the cingulate gyrus. So we have 4 pieces of information: the AV nucleus of the thalamus, where it goes, the cingulate gyrus, and where it's receiving input from the mammillothalamic tract and the fornix. Taking those 4 pieces of information, we now can very, very confidently say that the major system in the thalamus that is implicated in limbic, motivational, emotional types of function is going to be the AV nucleus of the thalamus. Why? Well, first of all, the limbic system, which was coined by the first head of NIMH, a guy by the name of McLean, who basically came up with an idea called the Triassic brain. And in the Triassic brain, he basically colored the brain in 3 colors. One was a deep, deep red. The second was a sort of pinkish color. And the third color was sort of sky blue. And the deep, deep red is what they would basically call a reptilian brain. And that basically, you're basically arguing here that whenever you're doing particular functions, especially eating, what the animal does is basically strike out. It basically goes, it hits, it's whatever. It's almost stimulus type of response, okay? Then what you have over it, the sort of pink area, is sort of an amphibian brain. So now think about our classic amphibian, the frog. If you went and you took 900 dead flies and put it in front of the frog had probably starved to death. The way a frog eats is, yes, it loves to eat flies and things, insects flying around either on top of the water or above the water. But basically what it's doing is it uses movement and stealth in terms of making its feeding response. And in this thing, this was called the sort of limbic brain. And then, of course, the blue is the cortex, which is a thinking type of brain. So what we basically have, and you can see this in so many models, is a model of pure, sharp, reflexive input, then of emotional responsivity, and then of cognitive responsivity. And then what you end up basically seeing is sometimes a conflict among the 3. So what are the areas? So the AV nucleus of thalamus was one of the areas originally described by the neuropathologist Papez, P-A-P-E-Z, in 1937, where he basically studied the histopathology of people with very, very severe and long-lasting mental illness, and found that the AV nucleus of thalamus was a site at which you would see some type of damage. And of course, the place to which the AV nucleus of thalamus projects, the cingulate gyrus, was also considered a part of the original limbic system. Now, where is the AV nucleus of thalamus receiving input from? It has the mammillothalamic tract. What is the origin of the mammillothalamic tract? Is the mammillary bodies that sits in the inferior surface of the brain in the caudal diencephalon. And that too was identified by Papez. Then the last one is the fornix. And the fornix, there are 2 areas that supply the fornix that goes into the AV thalamus. The major one is going to be the hippocampus, and the second is the septum. And McLean identifies the hippocampus and the septum as part of a little bit more modern limbic system. So what we can see is that the AV nucleus of thalamus seems to be very important in limbic types of functioning, and I'll talk about that pretty quickly. Now we move to the DM nucleus of the thalamus, or dorsomedial. And what we can see here is that the major inputs into the DM nucleus comes from the amygdaloid complex and comes from the temporal neocortex. And then the whole huge output of this very large dorsal tier nuclei is projecting diffusely up to the prefrontal gyrus, prefrontal cortex, all the prefrontal gyri, superior frontal gyrus, middle, inferior frontal gyrus. And what we can basically see here is a very, very important thing having to do with higher cortical functioning and interactions between the temporal neocortex and the frontal cortex in terms of a lot of cognitive tasks. Then we have the LD, or lateral dorsal, and LP, lateral posterior. Both of these are intrinsic thalamic nuclei. What do we mean by that? They don't have a clear, definitive extrathalamic input. As you can see, they're buried within this internal medullary lamina. So here we only look at the outputs of the LD and the LP. And the LD goes up to the cingulate gyrus, whereas the LP goes to the superior parietal lobule, which, if you remember, we talked about things having to do with associations, and I'll talk more about that. Then we get into the ventral tier. And the ventral tier nuclei, we have the VA nucleus. And then right after that, caudal to that, is the VL nucleus. The VA and the VL differ in terms of diffuseness versus specificity. And I'll explain that a little bit better. Also, they differ in terms of what their major input is. So here you can see the VA nucleus is getting inputs from the globus pallidus, which is part of the basal ganglia, and it's getting inputs from the substantia nigra. And most of this is the non-dopaminergic input. So the VA gets a globus pallidus input and substantia nigra non-dopaminergic input, and it projects out to the diffuse frontal cortex. The VL, on the other hand, gets information again from the globus pallidus and from the substantia nigra non-dopaminergic, but also gets input from the dentate nucleus of the cerebellum. And rather than projecting to diffuse areas of the frontal cortex, it projects to what is called Brodmann's area 4, which is, of course, our good old friend, the precentral gyrus. So you can start to see here that this is going to start to have input on eventual corticospinal activity. Then as we move, and what's interesting, look at how the thalamus is organized. The VA and the VL nuclei seem to be involved in either decision-making and motor and extrapyramidal motor things, whereas caudally in the ventral tier, we have the VPL and the VPM thalamus. So just like we had the precentral gyrus for motor and the postcentral gyrus for somatosensory, here what we can see with the VA and VL being more on the motor side and motor integration, whereas, of course, the VPL and the VPM are somatosensory. What are they doing? The VPL, of course, is getting information somatotopically from the medial lemniscus that, of course, are the axons of the nucleus gracilis and the nucleus cuneatus that, of course, receives somatotopic input from all 4 levels of the spinal cord. And then in turn, that the output of the medial lemniscus goes up to directly up to the postcentral gyrus in somatotopic fashion. And then the BPM, of course, is getting input from through the trigeminal thalamic tract. And it then sends a somatotopic projection out to the postcentral gyrus. Everything we learned about the somatosensory projection system. So now we move our attention. So you can see it's so much easier to say AV instead of anterior ventral and VA than ventro-anterior. You differentiate out. So then we have the 3 accessory thalamic nuclei. And of course, the first here is the medial geniculate body, where you're seeing the inferior colliculus through and the lateral lemniscus projecting to the MGB, and the MGB in turn projects out to the superior temporal gyrus for auditory processing. And then the lateral geniculate body receives ganglionic fibers, okay, that are coming in from both eyes into the lateral geniculate body, and then the lateral geniculate body sends fibers out to area 17. Excuse me, which is the primary visual cortex. So what we see is an enormous amount of inputs and outputs. It's not a place where all of a sudden the thalamus just says, okay, I'm going to send and act on some motor act. You have either somatosensory information, you have integrative information, you have motor information, you have limbic information, and then it's going somewhere else. So you're within a circuit which goes to make the argument about a relay. But relays usually just say, I began with something and then I hand off to something else. I don't change it. And the answer here is that's not the case. And the best example of this is the AV nucleus of the thalamus, which of course is within this limbic system. So very often we start out the limbic system. An easy way to start it out is to start with the mammillary bodies. And if you follow this, the mammillary bodies go up to the AV thalamus. The AV thalamus goes to the cingulate gyrus. The cingulate gyrus goes to the entorhinal cortex. The entorhinal cortex goes to the hippocampus. And then the hippocampus comes back and talks to the mammillary bodies. You have the proverbial circuit. Now that looks all wonderful if you're just believing in pure relay of this just going around and around, except for this little bizarre arrow right here. That is the hippocampus through the fornix acts on the AV nucleus of thalamus. So if for some reason the whole thing started with the mammillary bodies, the mammillothalamic tract basically innervates the AV nucleus of thalamus and then kicks off that so-called circuit, if that's how we believed it. But the AV nucleus of thalamus also is affected by the hippocampus through the fornix. So not only if we're basically saying the mammillary bodies is the beginning and the hippocampus is the end, well, look at what the AV nucleus of thalamus is getting. It's getting information from both the beginning and both the end, and this is integrating what the AV nucleus of thalamus does. So the AV nucleus of thalamus is interfacing in this large circuit, and in a lot of ways can either amplify it as a function of mammillary and hippocampal input, or diminish it in terms of the effect. Now, a very important kind of thing that we basically see is that the major output of the AV nucleus of thalamus and a major input to the cingulate gyrus is the input into the cingulate gyrus. And we have seen the cingulate gyrus, the vast size of it. And again, whenever we're looking at either cognitive functions, we're looking at emotional functions, we're looking at cognitive and emotional functions, and we're looking at this with fMRI, invariably the cingulate gyrus, and especially its anterior part, the ACC, the anterior cingulate cortex, invariably lights up. And now look at where the cingulate gyrus projects to. It projects to the entorhinal cortex. Now, where is the entorhinal cortex? It is predominantly in the temporal lobe. And look at its name, entorhinal. It is basically that part of cortex, although it reaches back more posteriorly, that has to do with what is called smell brain. And basically, if you're looking at animals exploring their environment, the sense of smell will give information having to do with mating. It will have to do with danger and aggression, and it will then have to do with approach. That is, so here is this animal tracking for food. It uses that type of information if it's predating, or it is looking for a sexual partner, or it encounters a conspecific in the area where there is competition, or it basically can tell that a predator that then eats it is nearby. And the entorhinal cortex, that is mostly around the parahippocampal gyrus, innervates the hippocampus and has a great deal of input upon the hippocampus. And then the hippocampus, then through the fornix, can go back to the AV nucleus of thalamus, but then goes through the entire diencephalon back to its path in the mammillary bodies. So this AV nucleus of thalamus is triggering all of these sort of cognitive / emotional aspects having to do with homeostatic behavior: eating, drinking, sex, aggression. Okay. So now we have the dorsomedial nucleus of the thalamus. And the dorsomedial nucleus of thalamus is receiving a lot of information from the temporal neocortex, but then also from the amygdala. Now, the temporal neocortex is described very clearly by a noted cognitive neuroscientist in the 1960s named Mortimer Mishkin. And basically, when he was studying hippocampal types of information, he basically divided the hippocampal information into two streams of output. One he called the dorsal stream of output, and the second he called the ventral stream of output. The temporal neocortex is interacting with the hippocampus and is a major player in ventral stream information. And in terms of learning and memory, what types of learning and memory might it be involved in? It is involved in semantic memory, the memory of events, episodes, words, whatever. And the second is episodic memory or event-related memory. Not only those single punctate objects, but how they relate to one another in some type of episode. And then, of course, in the rostral part of the neocortex, buried within there is the amygdala, which of course is a very powerful part of the limbic system and is very, very much involved in the production of what we call fear or apprehension, and then can be involved in a severe thing like phobias. So if you go back to your Psych 101 textbook somewhere, you'll basically read about two major classic theories about fear and how the amygdala might then be involved in that. And one is the James Lange theory, and the second is the Cannon-Bard theory. And this is the basic argument of chicken or the egg. So in James Lange theory, what basically happens is you have a whole series of neural events going on. Some of them are somatosensory, some of them are visual, some of them are auditory, a whole bunch of different events. And that you take each one of these events and you put it and synthesize it together, and you then basically come up with the whole kind of fear response. So it is that those series of independent sensory events that trigger an organism, let's say, to run, and then you basically then experience fear. Cannon-Bard is the opposite. Cannon-Bard argues that you integrate the thing into a fear message and then you run, okay? And these people just argue with adherents of both ideas, argued with one another for decades, and one could never solve the chicken or the egg issue. The person who came along with another group of researchers, was James Ledoux at NYU. And what James Ledoux basically started to do is he basically found that you could get these conditioned avoidance responses, a real fear response. You know, the animal will basically pull away with both an unconditioned stimulus and then with a conditioned stimulus. And then that conditioned stimulus will imprint and produce this fear response. He then started to do a whole series of lesions, especially of cortical structures, and found that you really didn't need those cortical structures to produce those fear responses, and any kind of change in the DM nucleus of thalamus was unaffected by the loss of those cortical structures. The critical structure that seemed to be important was the amygdala input. So basically, what the Ledoux's model is arguing is that this is a very, very powerful kind of emotional input, and that very often in certain types of situations, an organism may exhibit a fear response and then react without other types of cognitive input. this is an essential argument and underpinning for the argument of PTSD, where the amygdala is implicated, and where the patient may behave in an extreme emotional response in the absence of apparent cognitive cues. Now, where does this go? The DM nucleus of thalamus has mass projections all around the prefrontal cortex. And what it also does is reach down into the lower part of the telencephalon, a string of acetylcholine containing cells. And these cells will be anywhere from the outer part of the amygdala, the stria terminalis, to the nucleus accumbens, and it's a sort of a band of cholinergic cells. And this nucleus basalis of Maynard has a very interesting name. It is also called the substantia innominata. So what does that make me think of? It makes me think of the famous spaghetti Western movies that Clint Eastwood did called A Fistful of Dollars for a Few Dollars More, and The Good, the Bad, and the Ugly. These cholinergic cells, which are excitatory, and when activated by the DM nucleus of thalamus, produces wide-ranging activation of massive parts of the entire cortex, not only the prefrontal cortex. You're sort of galvanizing the organism. Why? Because you're getting from the amygdala emotional information, and from the temporal neocortex, you're getting semantic information and episodic information. And that is processed by the DM nucleus, that then activates on the prefrontal cortex, and then activates this large alarm system, a sort of cognitive enhancer system, which is the forebrain cholinergic system, which we then see severely compromised in Alzheimer's disease. Now, what's very interesting about this pathway, I can go on, there's probably thousands of studies that look at the prefrontal cortex, role of DM nucleus, but a series of studies were done by a highly influential female neuroscientist by the name of Patricia Goldman, and then Patricia Goldman-Rakich. When she was Patricia Goldman, she was down at NIH in the Michigan lab, and then she then married her husband, who's a prominent Yale neuroscientist, and joined him up at Yale. Now, she did a study that takes an enormous amount of patients and whatever. What she's basically doing is studying cognitive effects. And basically what you could find is in the prefrontal cortex, in a primate model, is that as you destroy the prefrontal cortex, what basically happens is you no longer get, there's no longer any input from the DM nucleus of thalamus, and the animal starts to suffer deficits in what is called a go/no-go task. And a go versus no-go task is you may have a light that comes on. If a light comes on, the animal goes and does something. If another light comes on, you stop from doing that. And it's very important to pay attention to both of the lights. It's sort of like that schoolyard game of Simon Says, and if you forget, you don't hear the word Simon Says, you don't do it, okay? So you're doing it or not doing it. And animals, adult animals with various losses of the prefrontal cortex show these enormous deficits in these go, no-go tasks. So what she basically did was ask, where in ontogeny does this happen? So what she basically did is produce a preparation in which she could take a pregnant monkey, remove the fetus from the womb, place a lesion in the prefrontal cortex or a control, put the monkey back in the womb, the monkey comes to term, now the monkey grows up. So now the monkeys have either a prefrontal cortex lesion of some size or not, and she now tests them on a go, no-go task. And what she basically found was that there was a subpopulation of the lesion monkeys that showed absolute deficits in their ability to do the no-go task, just like adult monkeys. And another subgroup that showed virtually no deficit. Now, when you're doing that experiment, what don't you have control over? What you don't have control over is what is the sex of that neonate. It could be male or it could be female. And she put males in the lesion group and the control group and female neonates. So now they grew up. And by the way, in adult monkeys, there's no sex difference. But what happened? She found out that male monkeys who received the neonatal ablation of the prefrontal cortex showed adult-like symptoms in the deficit of the go, no-go task. Whereas the female monkeys appeared to be resilient. They didn't show the effect. So now what we have to do is go into prenatal development and basically look at things. What we know usually in the first trimester is we are all in one way or another basically a female default. That is, there are factors, Müllerian inhibiting factor and Wolffian factor, that may produce, quote, male phenotypes and female phenotypes. So by the last trimester when she was working, you basically have the males and the females. In the males, you have a far more testosterone-laden brain because of the events that happen in the first trimester than in the females, okay? And so the testosterone, and it's shown in many, many other studies, that testosterone is a factor that will speed up this type of development. And what are we dealing with here? We're dealing with the prefrontal cortex, which brings us to the legal system, where there's an enormous degree of argument that basically says, especially when one deals with things like death penalty, and then deals with people who are under the age of 18 being sentenced to death because of a crime, because they were tried as adults, there's an argument that basically goes on that prefrontal cortex development in a human being can persist all the way into adolescence. And the same sort of thing can happen in the primate brain. So the point is, this testosterone-laden brain sort of predisposed this neonate when it suffered towards a final kind of formation of what the prefrontal cortex does. And then that animal losing the prefrontal cortex now has no other recourse and can't make the cognitive task. But what about the female? The female lost equal amount of prefrontal cortex, but she does not have, she has some testosterone, but certainly not the levels of a male. So what this basically had Patricia Goldman-Rakich do is take that data where you see this important sex difference and then basically ask what basically happened in the female brain that kept it resilient. And then when she basically looked at these prefrontal lesions in subsequent groups of neonatal animals and looked at them postnatally, the projection that ordinarily would go from the DM nucleus of the thalamus only in the female brain went and moved back to the superior and inferior parietal lobule. You literally, there was a resilience simply because testosterone in some ways timestamps and says, all right, this is the final destination, even though it gets destroyed. In the female brain, that female had more time so that these fibers could get redirected into the parietal lobe. So what you see in here is not only the importance of these DM nucleus thalamus fibers in these cognitive and emotional tasks, but you also see sex differences, and you see ontogenetic differences. And this really opened up an enormous area of other types of research. Patricia Goldman-Rakich was a distinguished professor at Yale. She was a former president of the Society for Neuroscience. And then a horror happened. She was walking around downtown New Haven, steps into the crosswalk with a green light, and all of a sudden a car comes plowing through, hits her, and kills her. But she's a remarkable kind of person who really trailblazed in a lot of ways this area. Okay, let me just stop here for a second. I want to see.
Dr. Bodnar, can I ask a question?
Sure.
Yeah, so with the sex differences, it had nothing to do with like estrogen levels. It was simply testosterone.
Yes, in whole bunches of subsequent studies, it was found that it was the relative testosterone levels being developed during the third trimester that did it. Okay?
Okay.
One other thing you can tell me, so I then don't have to stop, is what time is it?
It is 11:11.
Good. Okay. I just wanted to see where I was. Okay. So now here is the LD and LP nuclei. I'm not going to spend much time on it. Both of them have intrinsic thalamic inputs, and one of them projects to the cingulate gyrus, which of course would implicate the LD nucleus in some type of limbic function, whereas the LP thalamus projects to the superior and inferior parietal lobule, which has to do with basic association type of learning, both in terms of operant learning and classical conditioning. Okay, so now we go to the ventral VA nucleus, the ventral tear nuclei. And what we have here is that the VA thalamus, like the VL thalamus, is going to be what is called a sensory motor integrator. In a lot of ways, you can play all kinds of games with this. Now, if you're basically going to argue that the VA nucleus is a passive relay system, it just doesn't work very well, simply because of the level of modulation and where the inputs are coming in and how things are integrated before then. So the substantia nigra, as you learned in the midbrain lecture, has about 50% of its fibers that are dopaminergic, and the vast majority of those fibers project through the nigrostriatal tract up to the caudate and putamen, okay? And the substantia nigra nigrothalamic tract is a non-dopaminergic projection that goes not only to the VA nucleus, but we'll see in the next slide, the VL nucleus. Now, here, the non-dopaminergic input is influencing the VA thalamus. But whenever the substantia nigra is activated, it could activate those non-dopaminergic fibers and do this, or it can activate the dopaminergic fibers, then work on the caudate and the putamen. And we're going to see that this is receptor sensitive. What we're going to basically see is that within the caudate and putamen, there are going to be D1 receptors and D2 receptors that will respectively excite and inhibit the caudate and putamen. So there can be very strong modulation, differential modulation in the neostriatum by the substantia nigra. And then the caudate and putamen, the neostriatum, newer part of the striatum, according to phylogenetic rules, then controls the globus pallidus, which by default is also called the paleostriatum. Now, the globus pallidus is going to give rise to 3 fiber tracts. 2 of them we can see here. The ansa lenticularis projects to the VA thalamus, and the lenticular fasciculus projects to the VA thalamus. So, based on information from non-dopaminergic input of the substantia nigra, and based upon information coming from the globus pallidus, the VA thalamus is going to have particular types of firing patterns. And this pathway, the VA thalamus, then projects all over the diffuse frontal cortex, okay? So it's all up in the superior, middle, and inferior frontal gyri, and even into the precentral gyri, okay? Now, the diffuse frontal cortex, what we know about it, actually does indirect feedback on the VA thalamus through 2 ways. Number 1, the diffuse frontal cortex comes down and synapses on the substantia nigra, mostly through the corticobulbar tract. And second of all, the diffuse frontal cortex provides powerful excitatory activation of the neostriatum. So the neostriatum is activated, and the caudate and putamen will then come down and will then modulate the globus pallidus. So what you can see through these feedbacks is these feedbacks then modulate the ongoing activity of the VA thalamus, either through the substantia nigra or through the globus pallidus. Okay. Now we go to the VL nucleus of the thalamus. And the VL nucleus of the thalamus has a more specific type of projection. It's projecting right up to the prefrontal cortex. So it doesn't involve that much the input going into the precentral gyrus, but it's doing this input. And just like before with the VA nucleus, the VAL thalamus gets input from the nigrothalamic tract, or the non-dopaminergic projection, into the thalamus. And it gets input from the globus pallidus through those two pathways that we talked about before, the lenticular nucleus and lenticular fasciculus and the ansa lenticularis. And of course, the substantia nigra dopamine pathway modulates the caudate and putamen. The caudate and putamen in part modulates the globus pallidus. But then through the superior cerebellar peduncle, the cerebellum, and particularly the dentate nuclei of the cerebellum, will send a projection up to the VL thalamus. So what this is now getting is not only that ventral stream information, and it's also getting sensory motor integration, but it's also getting cerebellar input. Now, two things happen. That is, the VL thalamus and the prefrontal cortex are reciprocally connected to one another. So in addition, if you look with the VA thalamus, notice it's a one-way street. The VA thalamus just talks to the diffuse frontal cortex. The VL thalamus talks to the prefrontal cortex, but also gets information back. Further, just like before, in the diffuse frontal cortex, including the precentral gyrus, there is indirect input from the precentral gyrus. In the VL nuclei, there is direct input. The precentral gyrus, although it doesn't receive inputs from the VL thalamus, sends outputs. So the VL thalamus is controlled, whereas the VA thalamus is primarily controlled by two major structures, the substantia nigra and the globus pallidus. The VL thalamus is controlled by 5 structures, the substantia nigra. There's my pointer now. I lost my pointer. The substantia nigra, the globus pallidus, the dentate nucleus of the cerebellum, the precentral gyrus, and then feedback from the prefrontal cortex. Now, what is also important is the prefrontal cortex, of course, can reach back all the way back down and influence the substantia nigra. The prefrontal cortex can also, through the corticobulbar tract, affect the dentate nucleus of the cerebellum. So what you basically are having here is this very important loop. And then, of course, we then have the cortical involvement on the caudate and putamen that modulates the globus pallidus. So what you're basically seeing is an integrative type of loop here. And we're going to revisit this when we start to focus on another part of the brain, the basal ganglia, and then look at it from the lens of the cortex and the thalamus. So what we can end up with is 3 different versions of things. In a vitalistic argument, what we might basically say, vitalism saying there's something special about us, and since the thing that's special about us, all the attributes, also comes along with a big cortex, the cortex must be the boss. So we would then have a cortico-striato-thalamic loop, okay? On the other hand, we can look at it from the point of view of what's been around the longest, and that is the striatum. So we can have a striato-thalamo-cortical loop, or we can focus on the thalamus being the sort of place where lots of stuff comes in, and we can have a thalamo-cortico-striato loop. However you place the emphasis, even though you're talking about the same system, you're talking about different degrees of emphasis, and people have various theoretical ways of looking at things and whatever that, you know, argues here, okay? So now, here is something I hope we all remember and know. This is very important for you to keep in your head, is we're starting out here in the medulla, and we're looking at the nucleus cuneatus and the nucleus gracilis on both sides of the brain. And we recognize the input into the nucleus gracilis is somatotopically coming from sacral, lumbar, and lower thoracic inputs, whereas inputs coming into the nucleus cuneatus are primarily upper thoracic and cervical inputs. And then the second order nuclei, the outputs of the nucleus cuneatus and nucleus gracilis, are given two names. Before they decussate, we call these fibers, as they go around the central canal, the internal arcuate fibers. Then they cross the midline and then become the medial lemniscus. And they move through the medulla, through the pons, through the midbrain. And then somewhere in the medulla, a second fiber tract, the anterior spinothalamic tract, which is primarily coming from second order neurons of lamina 3 and lamina 4 that have already decussated across the anterior white commissure in the spinal cord, will join the medial lemniscus. And so therefore, what you have is, if this is the right side, you can see that because of the internal arcuate fibers and the decussation in the anterior spinothalamic tract, it is now represented on the left side, and information on the left side is now represented on the right side at the BPL thalamus. And the BPL thalamus keeps this somatotopic arrangement of input, and then the axons of the BPL thalamus comprise part of the posterior limb of the internal capsule and your synapse in the postcentral gyrus. Now, the BPM nucleus, when we started to talk about the trigeminal nerve, which we know is a mixed cranial nerve, and that we know that the sensory portion of the fifth cranial nerve is coming in mostly in the pons and the caudal midbrain. And this input, which is coming from the ophthalmic, mandibular, and maxillary branches, the second order neurons will form the trigeminal thalamic tract that basically innervates the entire head. And that goes to the BPM nucleus of the thalamus, and the axons of the BPM thalamus form through the posterior limb of the internal capsule and go to the postcentral gyrus. Okay, so that is somatosensation. Now we move to the lateral geniculate body and look at the visual system. So here is a quick showing of the human visual system, and it's marked by what? that because the eyes are forward facing, we get information from both the right visual field and the left visual field into both eyes simultaneously or near simultaneously, versus my friend the chicken, the right visual field is only seen by the right eye, the left visual field is only seen by the left eye. So the nasal portion of each hemiretina is getting information, the left eye, the nasal hemiretina is getting information from the left visual field, and the outer temporal hemiretina is getting information in the right eye from the left field, left visual field, and vice versa. So now the information goes through the ganglion cells, and now gangliofugal fibers that are formed out in the temporal hemiretina are going to stay ipsilateral. So the information from the right visual field will end up in the left temporal hemiretina and stay there and go back to the lateral geniculate body. Whereas information from the nasal hemiretina comes in and decussates. So what basically happens with this partial decussation, what basically happens is information from the left visual field from both eyes are going over to where? The right lateral geniculate body. Information from the right visual field going into both eyes will end up in the left lateral geniculate body. Okay, so now what we can basically see here is the various layers of the retina, and what we can again see here is the funniest thing is the very end edge of the retina. The furthest back part of the retina, of course, has the rods and the cones that is involved in phototransduction. And then we have an outer plexiform layer, an inner nuclear layer, which are the bipolar cells. And then we have the ganglion cells. But where are we actually moving? We're moving towards the front of the head. We're not moving this way, okay? So therefore, first of all, there's a miracle here as far as I'm concerned. How the hell that a rod cell or a cone cell picks up anything, because you have to traverse, the light has to traverse through the ganglion layer, the inner plexiform layer, the outer plexiform layer, and finally get to the retinal transductions. So here we can see in again the back of the in the back of the retina we have rod cells and cone cells, and of course most of the cone cells are going to be found foveally, whereas the rod cells can be found throughout, but in the extreme nasal and extreme temporal portions of the retina, you're primarily rods. These rods and cones then are going to synapse on two major types of cells in this inner nuclear layer. They're going to be synapsing on rod bipolar cells or cone bipolar cells. And then they're also going to be synapsing on horizontal cells. And it is the horizontal cells that are going to start to produce collateral inhibition. And the bipolar cells that, because of their connections with the ganglionic layer, is going to start to give you greater and greater acuity. And what do I mean by that? That for virtually every cone cell, there is a literal one-to-one relationship between an individual cone cell and its pair, the bipolar cell. Whereas the rod bipolar cells are getting inputs from a large number of rods. So the whole thing of saying, oh, there's something out there in the periphery is held in here because a rod bipolar cell will fire, but it won't know where it was fired from. So the acuity of something that you might have detected out in the periphery is not good. So when you're out there in the dark and you want to look at something, don't look at something foveally. First of all, the whole thing's of color, the whole thing's of contrast. Don't work in the dark simply because it's too dark an area. So what you should do is always look at the so-called corner of your eye. Okay, so now, and the rod and whatever rod and cone cells that fire will, of course, maximally affect horizontal cells that then go on and inhibit neighboring bipolar cells to focus the firing. Then the bipolar cells, through the interflexiform layer, now forms a whole series of different types of ganglion cells. There's P cells, M cells. This is beyond the scope of this course to talk about the function. But you're getting, with rod bipolars, again, there is sharing of ganglion cells. With cone bipolars, you're basically getting almost a one-to-one relationship. So now the axons of the ganglion cells now have to leave the retina. But remember, where is the back part of the retina? The back part of the retina is all the way up here, right? So what do these ganglion cell axons have to do? They have to go this way and come right back through this area. So there's going to be remember when we looked at the decussation of the corticospinal tract, where it almost ablated particular parts of the dorsal horn? Well, the same kind of thing happens here in the retina. Somewhere about 17 degrees off of the focal point of the fovea, there is a place called the blind spot. And that's the blind spot is because instead of having rods and cone photoreceptors there doing phototransduction, you have ganglion cells all packed together, myelinated ganglion cells, trying to exit the eye through the optic nerve and go back into the brain. So why don't we see a little blank space in our vision? Well, we have now done something very interesting to accommodate to that. What we have are things called saccades, S-A-C-C-A-D-E-S. And a saccade is a small bilateral conjugate eye movement. So this is all triggered by the visual cortex controlling the what? The frontal eye fields to execute these conjugate movements. So you're basically saying, even though your eye just moved a little bit, we know that the image is still in place, okay? So the outermost segments are involved in phototransduction. Here is, you can see your friends, the rods and cones, basically in the back of your retina, and that cones correspond to wavelengths of long, medium, and short. and is proportional to the intensity of light. So you get the blue, green, yellow, red. And then what you then end up with is rods that are not that sensitive to light. And here's the distribution of rods and cones. So you see far more cones in the foveal portion. And then down here you can see is the blind spot, okay? And here is the whole physiology of this. I just don't have the time to go through it, with on-center bipolar and off-center bipolar. And of course, a lot of this work derived from the doctoral dissertation supervisor of David Hubel. His name was Stephen Kuffler, and he was the person who sent Hubel to NIH, where he paired up with Wiesel, and they won the Nobel Prize. If they were going to give a logical Nobel Prize, they would have given it to Hubel, Wiesel, and Kuffler for Kuffler's work with on-center and off-center, and the whole idea of collateral inhibition, together with all of the contributions of Hubel and Wiesel, but Kuffler died. Okay, and then you have the morphologies of these cells, the horizontal amacrine cells. Again, lateral inhibition not only happens between rods and cones and their bipolar cells through horizontal cells, but they also happen between bipolar cells and ganglion cells with the inhibitory actions of amacrine cells. And again, here you start to get this whole idea of center surround inhibition. Again there, okay, there's the site types. So now we get to the chase, the anatomy. So like I said, the lateral geniculate body is sort of laminated. It has 6 different layers. And of course, you're now basically starting to take and you're starting to look at the visual field of the left eye and the right eye, and then see after this partial decussation, how this information then falls in the cortex, but also how it falls in the lateral geniculate body. And the way that this happens is the following. The fibers that do that partial decussation that come from the nasal hemiretina are basically going to layers 1, 4, and 6 of the lateral geniculate body. Whereas the temporal ganglionic fibers that are staying ipsilateral go to layers 2, 3, and 5. So of course, if you're following the image or the transduction, information from the left visual field is going to the nasal hemiretina that then of course cross at the chiasm, and information from the left visual field goes into the right eye temporal hemiretina. So it's a transduction of, one is a transduction of light waves, whereas the other is a transduction of visual information across the optic chiasm. So 1, 4, and 6 are the fibers that cross the optic chiasm from the opposite eye. 2, 3, and 5 are coming from the ipsilateral eye. Now, there are 2 types of information that happens at the lateral geniculate body, depending on whether you're looking at the ganglionic fibers coming in or the lateral geniculate body cells that are receiving them. What we basically find is that one retinofugal fiber that comes out of the ganglion cells can project to many lateral geniculate body cells in the layer you happen to be, whether it's crossed or uncrossed. And on the other hand, the lateral geniculate body is receiving many inputs. So now we can now go and divide up. We realize that the left visual field and right visual field, the left visual field, because of the partial decussation, is coming to the right LGB. The right visual field is going to the left LGB. But in addition, what basically happens when light enters the retina? It is inverted. So the superior visual field quadrant ends up in the medial LGB of either side, and the inferior visual field quadrant ends up in the lateral portion. And this happens whether we're humans, whether we're primates, or whether we are chickens. Okay, so if we basically look at this, we basically see that if a chicken is walking around and all of a sudden it gets wary about something predating it from above, you can see that chicken's medial lateral geniculate body firing like crazy. If, on the other hand, it's paying attention to whatever the hell it's pecking at, I'm just checking the time here. Oh, good. It's 11:44. If it's pecking the ground to figure where I'm going to get food, the lateral geniculate body can get activated in that chicken. Okay, so there is my little thing on vision. So now the medial geniculate body, and we're not even paying attention. You can put this slide together with the slides I presented to you earlier in the medulla, but here we're starting from the inferior colliculus, where we know by now what basically is happening. 60% of the input going into the inferior colliculus is coming from the contralateral ear, whereas 40% is coming from the ipsilateral ear. So we get, sound discrimination and all of these other kinds of things that can play a role here. And these inputs are coming into the inferior colliculus via the lateral lemniscus, and then the axons come out through the brachium of the inferior colliculus and synapse into the medial geniculate body. Now, if we basically go all the way back out to the inner ear and we look at the cochlea and look at the basilar membrane, the basilar membrane of the cochlea can be differentially sensitive to this to the amplitude of the transduced sound wave, and that is then is capped, and it goes through all of those crazy pathways involving the dorsal cochlear nucleus, the ventral cochlear nucleus, the superior olivary nucleus, the nucleus of the trapezoid body, and then coming up to the inferior colliculus, so that when we finally get to the medial geniculate body, and we look at the medial geniculate body and the medial portion of it versus the lateral portion, what we find is that the medial portion of the medial geniculate body, its activity is far more sensitive to high frequency sounds, whereas the lateral portion of the MGB is more sensitive to low frequency sounds. So now the axons of the medial geniculate body project through the geniculotemporal radiations, and then synapse again in a tonotopic manner to the superior temporal gyrus. And here we can see this auditory tonotopic innovation. So here what you have is this sort of a large array of microelectrodes that go across the secondary primary auditory cortex. And what can you basically see is that the rostral part of the rostral part of the superior temporal gyrus is very sensitive to lower frequency sounds. So of course, that rostral part is getting information from the lateral portion of the MGB. Whereas the caudal part, the mid and caudal part of the superior temporal gyrus is getting higher, higher frequency information. And that is, again, correlates with the base of the cochlea, and it correlates with the medial portion of the MGB. So basically, where the transduced sounds are happening either in the basilar membrane apex of cochlea or the basilar membrane base of cochlea, because this is all stretched out, corresponds to that part of the medial geniculate body that's going to get activated, and then counteract is with the superior temporal gyrus. So then all one has to do is look at particular sounds or sound range where you're basically looking at human speech, and you can then start to see where that is mediated as well. Then further, just like in the visual system, there is a columnar organization of both binaural and monaural input. Monaural input, of course, is input coming from the from one ear, and the other is where it's coming from two ears. So now think about this. A sound wave is coming directly at you and hits you right between, right at the base of the nose. So that sound wave is now going to travel around the head and enter each ear directly. The sound wave, and alternately, a sound wave comes in and hits you on the occiput of the head, right at the level of the bridge of the nose. And that sound wave is going to come around and enter each ear at the same time. Any other sound wave that's not either coming directly towards you or coming directly behind you is now going to hit, and especially a sound wave that comes in at 90 degrees, it hits the left ear, and some of that sound wave goes into the left ear, but it also travels around and now enters the right ear, or conversely, the right ear now coming in and conversely going into the left ear. So there is a delay depending on where the sound wave is in relation to the organism's head. So this is why, so if all of a sudden you hear a big bang out here, you will focus your head this way, simply because you have identified through binaural input, not only the original sound is coming in through this ear, but also the delay between the sound coming in this ear and the sound going in this ear versus over here. But what's very interesting is a sound can come in directly, well, in most cases, a sound may come in right towards you, and very often you might turn your head, simply because you interpret the sound as coming from behind, especially if the sound has no other kind of visual or somatosensory source. So what you can see, you have a different weighting between the two ears, and you have suppression columns where you're excited by one ear, inhibited by the other. And you have, again, just like in the visual dominance columns, you have an analog with the binaural, monaural input. So now it brings us to perhaps one of the largest thalamic nuclei, and that's called the pulvinar. And the pulvinar has, even though it has a massive size and millions of neurons, what it has is a very interesting reciprocal connection between the secondary and tertiary visual cortex and the pulvinar. The pulvinar doesn't go to primary visual cortex. It goes to secondary and tertiary visual cortex, and secondary and tertiary visual cortex communicate with the pulvinar. Now, here is an interesting thing in neuroscience. And we went all the way back to people like Cahal and Golgi, who gave us the very, very basic information about how the brain is structured and organized, yet came to very, very different conclusions. Whereas Cahal talked about neuron doctrine, that each neuron is doing something quite specific. It might be related to a neighbor or unrelated, but it's that individual neuron, versus the reticular kind of theory of Golgi, basically argued that a neuron and another neuron are probably not very dissimilar from one another. And it is the number of neurons, it's the engagement of sort of a crescendo, ascending or descending sounds that's going to produce this effect. Now, this also permeated into function. And the argument here would be that specific areas of the brain do something, but do they do it because individual neurons or individual groups of neurons within that particular structure do something that is highly specific relative to its neighbor? Or is it something, again, like mass action and something that you could call equipotentiality? By equipotentiality, it is the number of neurons in an ensemble that make the change in something versus which specific neurons do so. And one very powerful proponent of equipotentiality or mass action was a guy by the name of Carl Lashley, K-A-R-L-L-A-S-H-L-E-Y. And Carl Lashley was extremely well positioned back in the 1920s, 30s, and 40s, because what he was the head of was the Yerkes Primate Center in Atlanta, Georgia. So what he had was this enormous colony of monkeys. And what he did was elaborate, incredible types of studies that basically go and look at visual discrimination, visual perception, and then the neurobiological emphasis. So he would train all of these monkeys in order to get like the famous grape juice squirt into their mouth to do very fine visual discriminations, okay? So these monkeys would be trained over sometimes hundreds of days and everything. And then what he would do is he would do manipulations of the visual system. And one of the areas that he did this in was in the pulvinar, where he started to remove various parts of the pulvinar and various amounts of the pulvinar over a large number of monkeys. And you can just imagine that if you started to affect a certain part of the retina, or you lesioned a particular portion of the lateral geniculate body on one side or another, or you affected a very specific region of the primary visual cortex, you would come up with an argument about a very specific visual deficit. But here, what are we talking about? We're talking about a thalamic nucleus to pulvinar that gets secondary and tertiary visual cortex information. Information that in essence is arguing we have moved from a sensation, a visual sensation, to a visual perception. And when you lesion that, what he found is that it was the amount of the pulvinar rather than where in the pulvinar that predicted the deficits, and made a very eloquent and long-lasting argument towards some aspect of mass action working within the brain. So that basically makes me finish the thing. So I see people love Pavlovian fear studies. Love, that's an interesting verb for doing that. They're very powerful. And the fornix is a pathway between the hippocampus and the AV nucleus of thalamus, along with the mammillothalamic tract. And the fornix also projects to the hippocampus, from the hippocampus to the mammillary bodies, yes. And then what we're also going to see is that the fornix is a two-way street between the hippocampus and the septum, and we'll talk about that in a future lecture. So here we are, it's exactly 12 o'clock. We have finished the thalamus, and I hope by the end of this lecture, please don't think of the poor old thalamus as one of those second or third runners in a relay race. It not only relays information, but as we're going to continue to see, it integrates and amplifies that type of information. Okay, so with that, I'm going to say this is the end of the lecture because it's now 12:01, and I'm going to give you a break, and I'll see you all. Okay,
Thank you, Dr. Bodnar.
Bye-bye.
Bye.